We know that the cell is the basic functional unit and it is broadly classified into three parts. Number 1 is your cell membrane, number 2 cytoplasm along with its organelles, and number 3 is our nucleus. So we have finished the discussion of cell membrane in our class of cell membrane. Today we will learn something about the second part, that is cytoplasm and its organelles. Coming to cytoplasm, it is an intracellular material enclosed within the cell membrane and it surrounds the nuclear material, right? It is clear. It is present within the cell membrane and around the nucleus. And it is subdivided broadly into two types, that is number 1 your cytosol, number 2 is its constituents, that's nothing but the organelles. Now we are all familiar with the cell organelles, so let's quickly look at these. The organelles are important ones: mitochondria, endoplasmic reticulum, Golgi apparatus, ribosomes and lysosomes. In this session today we will be learning about each of them in detail. And of course the structural framework of the cell is maintained by your cytoskeleton, and for cellular transport there are something called the molecular motors. Okay. Now there are a few activities that occur in the cytoplasm. We all know these pathways and these shunts, so let's just look at these names. Glycolysis, that's nothing but your EMP shunt, then glycogenolysis, then the hexose monophosphate shunt, glycogenesis, fatty acid synthesis and cholesterol synthesis. Now what you have to understand here is the enzymes are present both in cytosol as well as the endoplasmic reticulum. The enzymes that are required for cholesterol synthesis are present both in cytosol and ER. However, major component is synthesized in the cytoplasm, okay, and there also occurs bile acid and bile salt synthesis. Now let's proceed to the important cell organelles. The first and most important thing that comes to our mind is your mitochondria. That's the powerhouse of the cell. So as we have discussed in our cell membrane class, the membrane is made up of a lipid bilayer protein membrane, right? This mitochondria is a cylindrical structure which has two lipid bilayer and protein membranes. It has two membranes, right? That is outer and inner membrane, and we are familiar with this word. It is described as the powerhouse of the cell. And why is it called so? Because of the presence of enzymes that are essential for the energy metabolism and the synthesis of the energy currency of the cell, that's nothing but your ATP, right? And it also, another striking feature is that it has an independent DNA apart from that which is present in its nucleus. It has an independent DNA and the functions importing and self-synthesizing of proteins. That's the function, and this is how your mitochondria looks, right? Now for our exam point of view, let's look at some important mitochondrial activity. So the important metabolic pathways are fatty acid oxidation, electron transport chain and the Krebs cycle or the TCA, or also known as the citric acid cycle, and also the oxidative phosphorylation. These are the pathways that occur. Now another important pathway, there are two pathways for which the initial steps they will occur in the mitochondria and the later steps occur in the cytoplasm. These are the urea cycle and gluconeogenesis. So part of it occurs in mitochondria and a part of it occurs in the cytoplasm, right? Now there are many enzymes that are present in various parts of the mitochondria. We will just have to memorize them. So let's look at them according to their location. Now in the intramembrane space or the intracrystal space, the enzymes present are creatine kinase and adenylyl kinase, right? Creatine kinase and adenylyl kinase. On the inner membrane of mitochondria, succinate dehydrogenase and ATP synthase are present. So ATP synthase, where is ATP synthase present? It is present on the inner membrane of the mitochondria. We have seen that there are two membranes, right? The outer and the inner membrane. So ATP synthase is present on the inner membrane along with succinate dehydrogenase. Now the mitochondrial matrix It has pyruvate and glutamate dehydrogenase and also enzymes required for citric acid cycle, beta oxidation of fatty acids and the urea cycle. All of these are present in mitochondrial matrix. The important ones that you have to remember here are the intramembrane space has creatine kinase, inner membrane has ATP synthase and mitochondrial matrix has glutamate dehydrogenase. Kindly try to memorize at least these three, right. So, the marker enzymes for mitochondria are the enzymes which are present in the matrix, the inner membrane as well as the intermembrane space. Okay. Now, let us proceed to the most important organelle after mitochondria, that is the endoplasmic reticulum. So, by definition, the structure of endoplasmic reticulum is an extensive network of tubules. It is a network of tubules running between the cell membrane and the nucleus, right. So, it is a network of tubules and we all know this, that there are two types of endoplasmic reticulum, the rough or the granular endoplasmic reticulum and the smooth or the agranular endoplasmic reticulum. Why are we calling this as rough or smooth? It is based on the presence or absence of the ribosomes. So, as you can understand, the smooth endoplasmic reticulum is devoid of ribosomes, hence it is called agranular or smooth type. Okay. Now, what is the function of smooth endoplasmic reticulum? See, smooth is for S, right. So, the first thing that should come to your mind is the steroid, right, steroid synthesis along with lipid synthesis, right. And in lipids, mainly phospholipids and cholesterol are synthesized in this structure called the smooth endoplasmic reticulum. Now, if I show you the rough endoplasmic reticulum, you can be able to compare these two. So, there is release of enzymes which influence the glycogen breakdown for energy production as well. Now, let us look at rough endoplasmic reticulum. Now, as you can guess, it has ribosomes, right, that are attached to its cytoplasmic site. So, these are concerned with the production of proteins, that is protein synthesis. This is how a rough endoplasmic reticulum looks and you can compare these two. Smooth endoplasmic reticulum does not have these small dots which are nothing but your ribosomes, right. That is why this structure is called rough or granular endoplasmic reticulum and it is the synthesis, it is the site of synthesis of proteins. So, it is involved in the initial polypeptide chain folding and disulfide bond formation. Two steps it is involved in: polypeptide chain folding and the disulfide bond formation. Now, let us look at the functions of rough endoplasmic reticulum. The first one we have just seen is protein synthesis. The second one we have seen a word called protein folding. Okay. This protein folding is done by structures called chaperones, right. They increase in quantity when the body temperature rises, hence they are also termed as heat shock proteins, right. So, protein folding is carried out by chaperones which are also known as heat shock proteins. Okay. Now, another important function of endoplasmic reticulum of the rough type is ERAD, that is endoplasmic reticulum assisted degradation. ER assisted degradation. So, we know that from this point we have understood the protein folding is also carried out in the RER, right. However, if the protein folding does not occur properly, that is if the proteins get misfolded or there are some toxic proteins, in these two cases endoplasmic reticulum assisted degradation occurs. Okay. That is ERAD. Now, what is the significance of this process is when such proteins, such misfolded proteins are not degraded, they lead to a disease called prion diseases, right. We are familiar with this word. So, they lead to the diseases called the prion diseases. So, if you look at this structure, this is the lumen of the endoplasmic reticulum and these are the misfolded proteins over here, these two. And what happens is when these misfolded proteins are present, we know that the IRAD complex along with ubiquitin causes degradation of these misfolded proteins by proteasome. Okay, this is the IRAD process. Now we have seen that they cause something called prion disease when there is no endoplasmic reticulum assisted degradation. And an important one over here is A beta amyloid formation also occurs when this process is defective, and this we know that it causes Alzheimer's disease. The deposit of amyloid causes Alzheimer's disease. So that is about rough endoplasmic reticulum. Now let's look at the functions of the smooth endoplasmic reticulum. We have seen S is for steroids. So the first one is synthesis of steroid hormone, and then storage of calcium, that is in skeletal muscle. The smooth endoplasmic reticulum of skeletal muscle is known as sarcoplasmic reticulum. Okay, that is the SER of muscle is called sarcoplasmic reticulum, and here its main function is storage of calcium. And then the third one is detoxification of drugs in the liver. Then smooth endoplasmic reticulum of the liver also stores cytochrome P450 enzymes. Okay. So the main functions are synthesis of steroid hormone, number 1, storage of calcium in sarcoplasmic reticulum, detoxification of drugs in the liver, and also storage of cytochrome